So now we are back with another topic, and this topic again comes under the chapter of the movement of the substances. And here we have to look on another important mechanism by which the substances they would move inside and outside of the cell. And this mechanism is entitled as an active transport. Earlier we have seen that there are total of the three major mechanisms that would move the substances inside and outside of cells. So we had the diffusion, then we have this active transport coming today, and it would move opposite to the diffusion. And then we have got this osmosis that would specifically be employed for the movement of the water molecules. See that the first thing that we have to crack is that what this active transport is. Now, as of before you have to see is that here we have got the particles, like there are more number of the particles. So any area which have more number of the particles, this would be regarded as an area of higher concentration. And if you were to look on toward the region B, so here we have less number of the particles. So where we have the less number of the particles, so this would be regarded as an area of lower concentration. So if I am to move these particles from an area of lower concentration where they are present in the less amount to an area of higher concentration where they are present in more amount, and that too if I am taking them against the concentration gradient, so this would be the active transport. But at the same time, something else that we must know is that when we have moved these particles against the concentration gradient, so we need to utilize energy that would be released by the mitochondria from the aerobic respiration, and there is an obligation that the cell membrane must be present as well. Likewise, when this active transport is being accomplished in the body, so along with the cell membrane, there would come some important protein molecules that would allow the particles to be moving from the area of the lower to the higher concentration. And these are the proteins, and the name of these proteins are the transport proteins. So if I move the particles from an area of lower to an area of higher concentration against the concentration gradient by using the energy from the respiration, by causing the particles to be moving across the membrane, and that too being utilizing the transport protein. So this is active transport. Now the reason is that why there has to be the involvement of energy. Now if I were to say that you have visited some hill station, and at that we have got this higher A point, and then we have got this lower B point with us. So if you were to fall from, or you were to come from the highest point to a lower point, then less of the energy would be utilized. But if you were to go against the gradient, so you have to be requiring more of energy. So same as the situation that is being accomplished here in case of the active transport. Now we must know that where in our body this active transport would be utilized. So see, when we would be digesting the food particles, and when I have to absorb them into the villi of the small intestines in the human body, here the active transport would be utilized. Other than that, we have this excretory system in our body, and when we have to be excreting the waste materials out of the blood, so it so happens is that along with the waste materials, some useful materials will also go out of the blood, so we need to be reabsorbing them back. So whenever in biology the word reabsorption is coming, so this simply means that something earlier was available in the blood, you took them out of the blood, and now you are taking them back into the blood. So here the mechanism employed would again be the active transport. So we have the importance or the significance of the active transport here in case of the digestive system and the excretory system. Now you see, you must remember as that where whenever in my body, if there would be the absorption of the food particles into the villi of the small intestine, so it would involve the active transport. Now our digestion is, and whatever diet is, that when we eat the food in our daily life, so we are eating complex substances, and these are the sugars or the carbohydrates, then we have these proteins, and then at the end we are eating the fats. So these are complex, they are large, and they cannot enter into the cells of the body until or unless we break them down or we digest them down into the small particles. So when digestion would take place, the carbohydrate would convert into the glucose, and the proteins would be broken down into amino acids, and the fats they would be converted into the fatty acids and glycerol. And this is happening where in the alimentary canal. But after we have broken them up, if I am not absorbing them into the blood, so this will be a problem. So that is why in the alimentary canal we have these villi where the active transport would be utilized, and these broken down food particles would be absorbed into the blood. Now for that I want you to look on toward this elementary canal structure that we have in the body. So you see at the top, what I have here, this is the mouth, and we have got an inner space here, and this is the buccal cavity. Then here would come the second part of the alimentary canal, which is pharynx, and then here is the stimulator structure, which we refer to it as esophagus, and here we have got the muscular bag, which is the stomach. Okay, guys, and then lastly, not the last thing, but here we have the small intestine. So we have to see the situation with the small intestine, because anything that I'm eating it up, so it would finally be digested into the small particles here in the small intestine. Then I have to be implying the active transport. So you just keep this thing in your mind, and now we would look on toward a bigger aspect of this part. And in the bigger aspect, you see that again this zigzag kind of pattern that you see here. So these are the same small intestines. And in the small intestines, now here we have this empty space coming. And this empty space would be regarded as a lumen, and in the lumen, the food particles, the digested food particles, they would lie here. Like I have shown you the glucose at this point, and I have shown you the amino acid at this point. Now we need to take them into the blood. And for that, we must know that how does these villi they look like? Because the villi are employing the active transport for the absorption of these digested food particles. So see, I have taken you from the small intestine, and in the small intestine, I have shown you the villi. Now we must look at what we have got in the villi. Now, in the villi, you see that these overall, they are the finger-like projections, like they look like finger-like. And we have multiple layers to be bothering about it. What makes this villi of the small intestine? So you see that we have this outer layer of the villi, and this outer layer of the villi has got these specialized cells in it. And these specialized cells that I'm marking with the yellowish marking, so you just remember that these are the epithelial cells. Okay, so this is one epithelial cells or the epithelial layer that I've got on the outside of the villi, and out of these epithelial cells, there would emerge small finger-like projections out. Like villi overall is finger-like projection, but it has got further, you could say projections, and these are the microvilli with us. Now, if I go from these epithelial lining to the inside, so here you see on the inside what we have, these are the blood capillaries where I need to take the food particles. And at the center of these villi at this point, now we have got something else, and they are referred to as electrials. So the situation is that outside of these villi, what we have got with us, the digested sugar in the form of glucose, the digested protein in the form of the amino acids, and the digested fats in the form of the fatty acid chains and in the form of the glycerol. Now I need to pick them up, and I need to pass them through the epithelial cells, and then I have to take them into the blood capillaries. Now, there lies something within these epithelial cells that would clear our concept that how come in the alimentary canal or in the villi of the small intestines, the food particles they are being absorbed. So you see here I have given the diagram of this particular epithelial cell, and if you were to see this, so here comes the cell membrane of the epithelial cell, and onto inside we have got these mitochondria, and then in the cell membrane of these epithelial cells, here we have these transport proteins with us. Okay, now we know that these mitochondria, what they gonna do, that they would release energy out of them by the respiration process. And that respiration process, when this releases the energy, so this energy would be utilized to move these glucose and the amino acids from the lumen of the small intestines, and they would pass through these transport proteins, and then they would come into the epithelial cells. And when they have come into the epithelial cells, from these epithelial cells, the particles they would move into the blood capillaries, and from the blood capillaries, these glucose and amino acids, now they would be carried to the cells where the cells they would utilize them for their cellular activities. So if I were to summarize this, so the situation has happened as such that we had the lumen. In the lumen, we have these glucose molecules, and along with the glucose molecules, these particles that we have with us, so they are the amino acids. Okay, then came this cell with us, and this cell, this was the epithelial cell of the villi, and it had some transport proteins as well in it. And then we had these mitochondria within them. When I came downward, so here we have these blood capillaries with us. Okay, I cast epithelial cells mitochondria to be releasing the energy, and then they move this glucose and amino acid with the help of the active transport, where they pass through the transport protein. They came here, and then from here they have moved into the blood capillary. So this is how the absorption of the digested food particles has happened, where in the small intestines of the alimentary canal with the help of the active transport. Now the second scenario was that whenever in our body we have to be bringing about the selective reabsorption of some useful substances. So this would be the selective reabsorption. The usual idea is that when multiple chemical reactions they would take place on the inside of my cells. So as a result of those chemical reactions, some of the waste materials they are getting produced, and among those waste materials that get produced as a result of these chemical reactions, so you may know the name of some. Among those, I have got something that is the urea. Then I have got something that is the uric acid. And after the uric acid, we have got another component, and this is creatinine. Likewise, from our diet, we would be drinking a lot of excess water. And that excess water, when we would drink, so there would be the presence of some ions, and these ions, they would be the excess ions with us. Now, my idea is that out of my blood, I have to take these waste materials. But when these waste material has to be taken out of the blood, so we have to rely on a system, and that system that we have with us. So this is the excretory system. Now, in the excretory system, we have got multiple of the things. Like we have these kidneys with us, then we have the ureters with us, then we have the urinary bladder, and then at the end we come to the urethra. So here you see this bean-shaped structure that I'm highlighting at the moment. So they are the kidneys. Out of the kidneys, these small tubes they are emerging out. So they are the ureter. And when I would come to the bottom, so here we have a muscular bag, and this muscular bag is the urinary bladder. And then here is this opening through which the urine would be released out of the body, and here comes the urethra. Now, this is the same way, like we have got the filter plants in our homes that filters the polluted or the dirty water. So likewise, if I have to take the waste materials out of the blood, so there should lie something within the kidneys where the filtration process must occur. Okay, and that filtration process, it happens in the kidney structures, and these kidney structures they are referred to as a nephron. Now here we have got the diagram of the nephron. If you look clearly, so we have this Bowman's capsule. Immediately below the Bowman's capsule, there comes this zigzag kind of a structure, and this zigzag kind of the structure is termed as the first coil tubule, or we call this as a proximal convoluted tubule. After the first coil tubule, if I'm gonna go downward like this, so here we have the loop of Henle. And if I come a bit above, so this is second coil tubule, and here we have the collecting ducts with us, or the collecting tubules with us. Now something happens here at the level of the Bowman's capsule where I need to filter the blood out. When I would be filtering the blood out, so not only the waste material but some useful materials would come along with it. If I were to rely that, okay, along with the waste material, the useful materials would also be lost in the urine. So this is a problem for my body because some molecules or some waste materials, we intentionally want to be releasing them out of the body, to be excreting them out of the body. But useful materials like the glucose and the amino acid, I can't afford. So here in the Bowman's capsule, we would filter the blood. And when useful materials would go along with some waste materials, so I need to reabsorb them back, and it would be accomplished with the help of the active transport. Now, the situation here is that this is the same diagram I have shown you. So you see that here lies the structure of the nephron, and this is a Bowman's capsule. In the Bowman's capsule, we have got these capillaries with us that are lying here, and they have the waste material and the useful materials. And these capillaries, they are entitled as the glomerular capillaries. In the excretory system, we will see that if the blood has to be carried into the glomerular capillaries, so this comes from an arteriole, and the name of the arteriole is afferent arteriole. And when I have to take the blood out of the glomerular capillaries away from the kidney tubules, so here we would have the efferent arteriole. E for efferent and A for afferent. Now the situation that is done here is that here we have got the blood in the glomerular capillary. This has some useful and the waste material as well. So I need to filter them out. When I would filter them out, so the situation happening is that in that way, I have taken the waste materials out and some useful materials have also come out. And among the useful materials that have filtered out along with the waste materials, now you must know the name for them. And the name are the glucose, and then we have these amino acids. Along with the amino acids, like we have got some ions that have filtered out along with the waste material. So I need to reabsorb all the glucose, I need to reabsorb all the amino acid, I need to absorb some of the ions and some of the water back into the blood. Because if these useful materials they would lie here within the nephron, within the structure of this kidney tubule, then it would be excreted out along with the urine, which is something I don't afford. So that is why I have mentioned at the start that in the reabsorption, something was already there in the blood. We took it out of the blood. Now we will take them back into the blood. So you see, earlier we have seen that here we have got this first coil tubule or the proximal convoluted tubule. And in this first coil tubule, there happens to be presence of some cell structures that would be utilizing the active transport, and the useful materials would be reabsorbed back. So now you see this is the first coil tubule with us. And I am showing you a bigger diagram of this first coil tubule. And in this bigger diagram, you see that here is the fluid flowing that we have filtered out of the blood, and it is termed as a glomerular filtrate. And in the first coil tubule, we have these functional cells with us. And these are the cells of the first coil tubule, or these are the cells of the proximal convoluted tubule. And if I were to show you a bigger diagram of these cells that we have got in the proximal convoluted tubule, so you see that in these cells, again, we have got their cell membranes, and in the cell membranes, we have got the presence or the incorporation of these transport proteins. And then again, in these cells, we have got these abundant mitochondria. So you now see from the structure that they are all employed, or they would all be used for the active transport. Now, here you must know that I have got this fluid flowing where there are waste materials and the useful materials. And among them, I have to reabsorb this all of glucose, and I have to reabsorb all of these amino acids so that they don't get lost in the urine. So what these cells of the PCT or the proximal convoluted tubule, they are going to do that they would employ their mitochondria. The mitochondria, they would release energy in the form of ATP, and that would be moving these useful materials out of the filtrate that we took out of the blood. They would come here into the cells of the PCT or the first coil tubule, and from there these useful materials they would be taken back into the blood. So here again you see that when we have reabsorbed the useful materials out of the urine or out of the fluid that is flowing through the kidney tubules, so the active transport has been utilized. So here we are done with the influence or the significance of the active transport in case of the animals, where we have seen in the digestive system and in the excretory system. For the digestive system, you must remember that we have absorbed the digested food particles, and for the excretory system, we have reabsorbed the useful materials or the useful particles that we were not in the position to be getting them lost into the urine. Now, the other situation is that we must know that how come this active transport this would be accomplished in case of the plants. Earlier in the specialized cells section of the plants, we have seen that the root hair cells were lying, and these root hair cells again they have abundant mitochondria, and they would be having these transport proteins that would allow them to be absorbing the ions. Now this structure should come in your mind in context of the active transport in case of the plants. So for the root hair cell on the outside, what we have, this is the cell membrane with us, a cell wall with us. And on the inside of the cell wall, we have the cell membrane with us. And here we have got at the center the presence of a large vacuole. The situation here is that the large vacuole already has got more of the ions in it, more of the charged particles in it, and on the surrounding soil that we have here, so here the ions they are present in less number comparative to the large vacuole. So within the large vacuole of the root hair cells, the concentration of the charged ion is more, and in the surrounding soil the concentration of the charged ions is less. So we need to be moving when we need to be moving these charged particles from an area of lower concentration in the soil to an area of higher concentration within the large vacuole of the root hair cell. Again, the active transport would be utilized for this purpose. Now, within the cell membrane of the root hair cells, what we have got are these transport proteins. And then within the cytoplasm of the root hair cells, we have got these abundant organelles. and these abundant organelles, they are the mitochondria with us. So these mitochondria, they would release energy by the respiration in the form of ATP, and that ATP would be utilized to move these ions in the lower concentration in the soil to the higher concentration in the large vacuole, and that is also employing some transport protein, and the movement is done against the concentration gradient. So this is how the ions, they are absorbed in the root hair cells with the help of this active transport. So how you have to remember as, or how you need to recall this as, you will tell your examiner that in the soil, the ion has lower concentration. In the large vacuole, the ions has higher concentration. The mitochondria would release energy in the form of ATP, and that would move the ions from the soil where they are in the lower concentration to the large vacuole where they are in higher concentration. And in doing so, the transport protein, and against the concentration gradient, we have moved them up. I hope you will be clear with active transport now. So see you in the next class.